Epigenetic Sex-Specific Fetal Alcohol Brain Deficits by Kathy Wilson. Spatiotemporal gene expressions is an accepted solution for new research for many fetal growth abnormalities. The debate continues into resilience, or the ability to overcome environmental stress or adversity during fetal formation. Chen et al., 2016. Prenatal alcohol exposure has long been known to be a teratogen and is identified as a fetal epigenetic environmental teratogen, and its adverse environmental impacts on primary fetal biogenesis prove a likely biomarker identifier for alcohol-exposed neonates. Barrett and Salzman, 2016. Newer high-quality resting state magnetic resonance imaging (fMRI) measures allow non-invasive fetal in vivio research, creating new understandings and clues, advancing knowledge. Anderson and Thomason, 2013. It is suggested that epigenetic developmental changes encourage a resilience susceptibility based on particular fetal genetic makeups, indicating a phenotype needing further study. Barzilay et al., 2020, Smith et al., 2014. Current research identifies the effect of PAE on fetal neuronal hippocampal migration, which relates to fetal reduced brain size. Wilhelm and Gazzetti, 2016. The second trimester is a gestational age, GA, period in which hippocampal function actively and aggressively associates with cerebral malformation. Alarcon et al., 2015. G et al., 2015. The asymmetric sex-specific effect on the developing brain infers alcohol's pathogenetic influence on fetal brain neural development and dysfunction difference between males and females. Gainsetti et al., 2020, Rugrock et al., 2014. In summation, a recent proposal calls for a precise and definitive fetal brain mapping of alcohol-induced altered structure-function relationships for use as predictor biomarkers for future action. Batista Garcia-Ramo and Fernandez-Verdicia, 2018. The benefit of using an MRI diffusion tensor imaging, DTI, is that this technique is recognized as a standard to investigate fetal neural pathways. Anderson and Thomason, 2013. The proposed quasi-experimental research will identify independent variables from in vivio investigation. The proposed study uses statistical designs for causal inference, which are quantitative, with statistics to estimate the causal effect, but require qualitative procedures. Magnetti et al., 2015. The attributive independent variables of PAE-exposed fetuses compares with a similar normally progressing fetal GA control group. An MRI diffusion tensor imaging, DTI, will visualize the fetal GA brain, with image slices mapping brain structure impairment and function. Slices with two-scale images and calculations creates a fetal map diffusion template. Batista Garcia-Ramo and Fernandez-Verdicia, 2018, Kahn et al., 2018. A GA fMRI capture and a DTI image visualizes brain function connectivity and white matter volume. Anderson and Thomason, 2013, Batista Garcia-Ramo and Fernandez-Verdicia, 2018. The blood oxygen level dependent, BOLD, is a brain measure for in vivio neuronal activity. Wheelock et al., 2019. A visual FSL viewer measures prefrontal cortex, amygdala, and hippocampi with a two-value gender comparison. PAE fetal characteristics include white matter myelination, abnormal brain plasticity, and reduced neuron survival. Wilhelm and Gazzetti, 2016. A resting state MRI images each fetal brain element in pixels. The mean gestational age criteria are weeks from the last menstrual period, crown to rump length, head circumference, and foot length. G et al., 2015. The dependent variables are physical traits, gender, and neurobehavioral indicators compared with the control group. A correlational analysis with sequential and multiple regression analyses of physical traits, gender, and physical indicators, and multiple sequential regression compares covariates of daily and binge drinking criteria, GA of the fetus, and maternal age in years, with a separate sequential regression by gender. Statistical analysis will segregate by sex across groups by one-way analysis of variance, ANOVA, and Dunnett's C-test will compare differences between groups. May et al. 2017. Hypothesis. The male fetuses diagnosed in vivia with PAE exposure will demonstrate sex-specific regional brain volume differences in the prefrontal cortex, amygdala, and hippocampi areas compared to females. To maximize the data relevance of the study, a strength of 100 intended participants will have similar GA characteristics. Given 2010. Participants are 30 to 37 weeks GA in vivio, and recruitment is at the Maternal Fetal Medicine Center with at-risk pregnancies and longitudinal study in Buffalo, New York. John R. O'Shea Children's Hospital, 2021. GA and criteria for addition into either group is determined by the study physician. The control subgroup and study group have similar GA from ultrasound analysis 1 week before the fMRI. Wheelock et al. 2019. The gestational age is weeks from last menstrual period, age of female in years, maternal drinking patterns, and fetal gender. Inclusion criteria include pregnancies with no contraindications for a resting imaging fMRI study, Batista Garcia Ramo and Fernandez Verdecia, 2018. The proposed study uses the University of Buffalo Research and Economic Development Research Center with procedures approved by the University of Buffalo Human Investigative Committee. All potential study participants will receive a consent document after education. Appropriate training for personnel, staff qualifications, plus possible risks to mother and child are explained before the fMRI imaging. Wheelock et al. 2019. Research personnel require additional specific training before performing fetal imaging to reduce stress association during the scanning process. Females naturally progressing pregnant volunteers at exit interviews highlight that with proper preparation for participants, staff, and researchers, time spent proactively creates a safer atmosphere to highlight protocol's positive, beneficial aspects. Van de Heuvel and Thomason, 2016. The fMRI is captured on a Siemens Vario MR system. The resting state fMRI data acquisition uses an echo imaging sequence. Each fMRI individually captured data is between 12 to 24 minutes of fetal resting state. Manual preprocessing will identify by FSL image. Then fetal brain masses are created individually for each image. Wheelock et al. 2019. After masking, each temporal segment is reoriented manually to determine the mean bulb volume, and then reoriented and standardized to a 32-week fetal brain template using JMP Analytics, SAS Institute 2021. Motion parameters for each manually identified inactive frame include data that has 10 frames of low motion rotational movement. Wheelock et al. 2019. Brain atlases are built from fMRI's fetal templates specific to GA and consider resting state volume slice motion corruption of images. The study solution uses the two-step reconstructive matching repetitious design to solve this error. Carter and Shea, 2015. The independent variable factors examines pregnant women defined with moderate to heavy drinking habits. The inclusion criteria defines moderate to heavy drinking as 2 or more binge drinking episodes, drinking standards as 1 12-ounce glass of 5% beer, cider, or cooler, a 5-ounce 12% glass of wine, or a 1.5-ounce shot of 40% hard liquor. Popova et al. 2020. Statistical analyses utilize the SPSS 27 IBM 2020 using independent t-tests from GA pinpointing brain regions of interest, ROIs, at the time of the scan. A t-test statistically compares the control group with the target group with normally distributed data. A two-way analysis of variance tests the association between DTI and fMRI, comparing brain matter weight for study control groups and gender. The control group will compare to the PAE group using a paired t-test to analyze normally distributed data. Kraska, 2010. New imaging techniques opens preventative in utero possibilities as neuroscience techniques advance science to a new level of hope, encompassing rehabilitation before birth. Albury et al., 2021. With novel neurobiological advances in the last 10 years and dedicated research topics that include PAE as a 100% preventable cause of neurological developmental disorders, PAE-exposed births can decline for future generations to ensure healthier outcomes across many lifespans. In 2017, Kahn et al. 2018 constructed the first spatiotemporal DTI atlas of the fetal in utero brain from the largest study sample then available. This is the first atlas for normal fetal brain development, which can be used as a reference guide for future abnormal fetal studies. I have included the DTI parameter atlas from Harvard at the end of the conclusion, and it is an exciting development for future research and may make in vivo epigenetic brain modification a possibility for future fetal interventions. Fetal Brain Atlas, Computational Radiology Laboratory, 2018.